welcome everybody. It's really nice to see so many of you. I see 55 participants, which is really great. I want to welcome you all to the Alberta Summer Series, Alberta MS Network Summer Series. By way of introduction, the Alberta MS Network is a collaboration across the universities in Alberta, and that includes University of Alberta, Calgary, Lethbridge, as well as Mount Royal University. And the collaboration is around training, and particularly training the next generation with the best tools possible. And also, the network seeks to facilitate collaboration across the province. It also attempts to bring in funding to fund initiatives such as summer studentships that a number of you are recipients of. So that's what the Alberta MS Network does. I'm the director. Neetu Singh, PhD, is the manager. And we have a steering committee across the province of Alberta that facilitates our activities. So this is to welcome you and also to tell you that this summer is like no other summer, as all of you know. Usually at this time, many of you would already be in labs, mingling with other summer students and also other trainees, as well as faculty across the different universities. But due to COVID-19, this is a different summer. So unfortunately, you'll be losing out on that intimate interaction that really is the exciting part of the day besides doing the experiments. Nonetheless, though, we'll do our best. Many of you are already well set with your respective supervisors on day-to-day reading and hopefully doing some experiments, at least online anyway, analyzing hopefully the images that have been collected, etc., etc. So we'll still do the best we can. And in terms of the Alberta MS Network, we are offering this weekly series through to August to bring you up to speed on many different areas of MS. And I want to thank the faculty for agreeing to lecture. The faculty comprises of some fairly seasoned lecturers, some newer faculty members, a number of postdoctoral fellows, and also some graduate students, all of whom are, I would say, very well informed. So we're looking, therefore, to a very exciting summer series. So the process is also that, for each Thursday, beginning at 10 a.m., there is a 1-hour lecture. Hopefully, we won't go too long over today with the audio-visual hiccups at the beginning, but slated to be for an hour. We would like you to make notes, ask questions at the end. If you have any questions at the end, just type in those questions and we'll try to get through them. We are hoping that at the end of, sometime in August, or I think sometime in August, that we can set up some special sessions with a number of you to deliver what you have learned and done over the course of summer to your colleagues, your peers, as well as to many of the faculty. So hopefully you will be motivated to share your results in August, and we do look forward to what you will be producing despite this being a challenging summer. I should also say that many of us will make our slides available in the form of podcasts through the Alberta MS Network. So this I think will be recorded. Neetu is recording. My slides can be shared with any of you if you approach Neetu for that. Okay? All right then. So without much ado, we'll begin. I thought we'll start with this first lecture around the disease state of MS. What is MS? What are some of the highlights? What are some of the pathology? What are some of the thinking around causes of MS? Next slide, please, Neetu. Okay, so here I thought I would schematize for you some of the players in MS. We have the central nervous system, the brain and spinal cord, that, as you know, contain billions of nerve cells that are referred to as neurons. And one neuron communicates with another and with many within the central nervous system. And of course, they do so by sending impulses down one nerve cell to another, where transmission occurs across synapses. Some of these nerve fibers or axons are very long in the spinal cord. They could be over a meter in length. And so because of that, we need mechanisms in order to facilitate the fast transmission of impulses. And that mechanism is enabled by these wrappings that are referred to as myelin, formed from cell type called oligodendrocyte. So here we depict that. Here we depict one oligodendrocyte forming one segment of myelin on an axon, but one oligodendrocyte can actually form sometimes over 50 myelin segments, usually located on different axons. So the oligodendrocyte is an important cell type that does a lot of work for multiple segments of myelin. And then we see in the blood compartment immune cells that circulate, and I show you an example, one example, a lymphocyte. And normally a few of them can enter into the brain and spinal cord by crossing this barrier, the blood-brain barrier, because they are necessary for immune surveillance of the central nervous system. But in normal circumstances, there are only a few of these that enter into the CNS. In multiple sclerosis, we'll see that many of these immune cell types are activated. They gain a pro-inflammatory characteristic. They gain the capacity to cross this blood-brain barrier that normally excludes them. And within the central nervous system, they then begin to produce injury. So here are the main players then in multiple sclerosis, and we're going to go into a little bit more depth in the next slide. Neetu, I don't think I can control my slide. Yeah. So here's a rendition which is captured from an MS brain specimen. We have the capacity to collect these brain tissues in a number of centers around the world. And from these tissues, we can cut sections, we can perform immunohistochemistry, for instance, or other types of stains to identify the various features. And here is a very magnified image of a blood vessel from which immune cells have now crossed that blood-brain barrier into the CNS parenchyma. So this just shows an example of a situation in a multiple sclerosis lesion where many immune cells have crossed into the CNS parenchyma and they will produce injury. Next slide, please. Neetu?
Sorry, it's not allowing me to. Wow, what's happening? Sorry,
Why don't you go?
Yeah, now.
Okay, good.
Now, yeah.
Okay. Now, so when these immune cells have entered into the central nervous system, they can produce injury. Here is another section of the brain. Can you see my cursor or not? Not really, huh? Okay, never mind. So here's a part of the central nervous system that contains myelin. Myelin can be picked up by this chemical called laxofast blue that stains the lipids of myelin and turns that blue. So you see normally in an area containing myelin, you should be seeing blue uniformly across. But as the myelin gets destroyed, when the oligodendrocytes die as a result of the immune cells accumulating and producing injury, the myelin is lost. So when you use this stain, there is now less myelin to pick up that laxofast blue, so that areas that have lost myelin would not be blue, and that would be an area of demyelination. So you see in the top left an area of spared myelin, and within the center, a demyelinated plaque that normally would have been blue due to the laxofast blue stain, but because it has lost the myelin, it is now no longer blue. Next slide, please.
What's happening?
And I'm sorry for this difficulty. Now, some of these demyelinated plaques can be quite large. And here's an example of an actual specimen from an individual that died with multiple sclerosis in Calgary. The brain has been taken out. The spinal cord is looking at you at the bottom. And normally, because myelin contains a lot of lipids, and lipids, as you know, are somewhat whitish, whereas the spinal cord should have been quite whitish, if you like, an area that has lost substantial amount of myelin that contains lipids and has therefore lost a lot of the whitish lipids, now looks less white, and that's a plaque that you see in the spinal cord. And often at times, many of these plaques are very large, and you can see them with the naked eye. Next slide.
Can you do it, V? I have given you controls.
You have given me control. Okay, okay. So let's try that. So why do we need myelin in the first place? So here I'm showing you an axon that conducts. I am afraid I am not able to. Can you do a click? Need to? Okay, so apologies. So what I was meaning to show you is this impulse that go down that axon. And next slide, please. Next click. Here's the situation where the same axons, same diameter, but now with myelin on it, is conducting an impulse. And next click, please. And what is supposed to happen is that you're going to see that impulse propagate along. It's not really doing that, unfortunately, but the presence of the myelin allows the impulse to rapidly spread down a nerve fiber. Next click, please. So apologies for that. Besides the loss of myelin, we want to emphasize that axons and neurons are also lost substantially in multiple sclerosis as well. Previously, I showed you the slide of the loss of myelin picked up by Luxofast blue, where an area of demyelination is not blue. And the demyelination can be easily picked up by neuropathologists. So for many, many years, MS was thought to be a condition of loss of myelin. But now we know that axons and neurons are also lost substantially. And indeed, it is the loss of the axons and neurons that are thought to be more critical in the disease manifestation. It is the progressive loss of axons and neurons that correlate better with the progression of disability in MS. So the myelin is lost, and that's important, but it is important to know that the axons and neurons are lost substantially as well, and may be more critical to the disease manifestation, because we need axons to conduct impulses. And here I'm showing you examples of loss of axons and neurons. The top panel shows you a controlled spinal cord in which the axons, which are circular in this case, are coming out at you at 90 degrees. And in a situation, in a plaque from an individual with multiple sclerosis, you appreciate that many of these axons are lost. In panel B, in living individuals using MRI, brain scans of the caudate nucleus, measuring the volume of the caudate nucleus in a group of normals and a group of MS individuals, you see that there is substantial loss of volume of the caudate nucleus, atrophy, and similarly shrinkage of the hippocampal volume as the disease progresses. Next slide, please. And this is just to depict for you that we have the oligodendrocyte forming myelin segment, that in MS, the oligodendrocyte is lost, demyelination occurs. The dropout of the axon is also a very important feature of the condition as well. And depending on the different MS cases, there are some MS cases in which the axons and neurons seem to degenerate first, but more often than not, one finds cases in which the myelin and the oligodendrocytes are lost first and the axons degenerate subsequently. Next slide, please. So that MS is not only a demyelinating disorder, but a degenerative one, and emphasis is that the continuous loss of axons and neurons is thought to be the principal contributor of progression of disability. Next slide, please. So they are no longer going as PowerPoint slides. I need to. Never mind. What this shows you is that over the course of multiple sclerosis, because of the degenerative processes, you lose many parts of the central nervous system shrink, and you have progressive brain atrophy. I should also tell you that lesions can occur in different areas of the central nervous system. More often than not, they appear around the ventricles, the holes that you see in the center of a brain scan. There are certain areas that seem to accumulate more lesions, but in general, lesions can occur throughout all areas of the central nervous system. And depending on where they occur, that will dictate the manifestation of symptoms. So if one has a lesion in the optic nerve, for instance, one would have disturbance of visual functions, for instance. Next slide, please. What causes the neurodegenerative changes? They're caused in part by inflammation. Next slide. And the reason for thinking that is based on a lot of evidence, including an example like this, in which you have an active plaque. And again, this section has been stained with Luxo-Fast blue. Everything should be blue in the background if this is an area containing a lot of myelin, but you have a loss of myelin, the demyelinated area, and one shows you a vessel with those dots there. Those are immune cells that are crossing the blood-brain barrier into the CNS, and where they accumulate in the CNS, those are areas of demyelination. So an area of injury occurs in an area of cellular infiltrates. Next slide, please. And I apologize for this, but the painful process, all of you. We'll sort out this next week. And it's probably because I don't know how to control slides from my laptop. But nonetheless, I hope you are still following along. Here is now, again, trying to emphasize for you an area of injury, an area that is actively undergoing loss of myelin, demyelination, and that area is correlated with where the immune cells are infiltrating into the CNS parenchyma. So you have images here that are progressively zoomed out, if you like. So you have a ×40 objective image in which you have that blood vessel in the middle. You see the red blood cells directly in the middle. You see those dots, immune cells coming out. And as we progressively zoom out, you'll see that areas where those immune cells are getting lighter and lighter in terms of the blue, because the myelin has been destroyed. And if you look at the ×5 image at the bottom right, you see lesions of different ages. Right in the top right is an older lesion because much of the myelin has already been lost. And then you see in the middle strip, more recent injury because it is still somewhat blue, whereas in the bottom left, the blue is intact. That's still an area that is relatively preserved at this point. So data sets like this, as well as other data sets, lead us to lead us to the conclusion that while we still do not know what sets MS off in the first place, what we do know is that certain immune cell subsets get activated. They gain the capacity to cross the blood-brain barrier to enter into the CNS parenchyma, and where they gather, they can bring destruction, and that inflammation, however MS starts in the first place, is one of the contributor to the degenerative processes. Next slide, please. So in terms of what types of immune cells enter into the central nervous system, we can analyze that by using immunohistochemistry, for instance, using antibody, in this case, to CD3, which is a marker on T cells. So those brown cells are T cells because they stand for CD3. And then next slide. So we do know, therefore, that T cells enter into the CNS. And here in this next slide, we're using a stain for CD68 that is present on macrophages that, of course, come from monocytes in the bloodstream. So circulating monocytes, when they enter into tissues, are referred to as macrophages. But we also have CNS intrinsic immune cell, which is called microglia. And here one shows that CD68 that picks up both macrophages and microglia, that lesions in MS are actually quite abundant with activated microglia as well as macrophages that have come from monocytes in the bloodstream. So we have a number of immune cell subsets that get in the central nervous system, T cells, macrophages from the bloodstream, microglia within the central nervous system. We'll also learn about astrocytes, CNS intrinsic glia that also gets activated. B cells are also an important component of lesion formation in MS as well. Next slide, please. So what brings about all of these? Well, we have skipped one slide, no problem. So here's, we may not be able to show, this is supposed to be a video. I don't have control of it. This video is supposed to show us a rendition of a simulation of what could occur in the central nervous system. This is a tissue culture experiment in which we have grown neurons, and you're seeing that long strip of axon, if you like, in tissue culture, to which we have added activated T cells. And you're supposed to see, if the video had worked, that these T cells are engaging that axon and eventually transecting that. So next slide, please. So a reminder on overview pathology of MS, overactivation of certain pro-inflammatory immune cell subsets, certain because not all of them get activated, that they then gain the capacity to cross the blood-brain barrier, and where they accumulate, they can bring with them potent neurotoxic components that lead to the neuropathology, the myelin and oligodendrocyte loss, and also the axonal and neuronal loss. And because all of these are very important for efficient conduction of nerve signals, of impulses from one neuron to another to different parts of the body, therefore symptoms arise. And again, depending on where these lesions are formed, this would dictate the spectrum of symptoms that you will hear next week from Dr. Penny Smythe. Next slide, please. So basically then, here is my overview, just to put it in a cartoon form. Normal situation, in the top one, very few immune cells enter into the CNS to perform immune surveillance in MS. Certain pro-inflammatory subsets get overactivated. They increase in numbers, they gain the capacity to cross the blood-brain barrier, and they produce injury within the central nervous system. Next slide, please. So what causes all of this in the first place? Well, the initial trigger is still unclear, and it may be different from one individual to the next. There are hypotheses of infections, particularly Epstein-Barr virus infection, causing certain immune cell subsets that can identify endogenous proteins, so potentially autoreactive T or B cells. That there's the hypothesis that viral infection can lead to the inappropriate activation of those potentially autoreactive T and B cells. There are also hypotheses of certain chemicals in our environment being able to activate immune cell subsets in an inappropriate manner as well. It's very, very difficult to prove causation in humans because we still have uncertainties as to when that trigger is encountered. Could it be during embryonic life? Could it be during early childhood or later on in young adulthood, for instance? All of those are unclear. So with that, it is very difficult to prove causation, but those are some of the hypotheses of something out in our environment causing the inappropriate activation of autoreactive T cells. We do know that genetics play a role in the development of MS. Looking at this data set from actually Canadian contributors, Dessa Sadovnik at UBC and George Ebers, who went on to Oxford University, what they have described is that amongst Canadians of European descent, the prevalence of MS is 0.1%, 1 in 1,000. If one has a twin with MS, a dizygotic twin pair, the risk of the other developing MS increases to 4%. If one has a monozygotic twin with MS, the risk of the other developing MS increases to 30%. But what you do know is that even amongst identical monozygotic twins, the prevalence is not 100%. So genes cannot account entirely for multiple sclerosis, and that's why there is a role for environmental factors. But following whatever exposure, immune cells get activated, and therefore the treatment of MS subsequently involves trying to tame the immune system. And again, you will hear about that next week. Next slide, please. So this is a recent publication on the genetic basis of MS by looking at the DNA of 47,000 individuals with MS, 68,000 of controls, many of which are spouses of those afflicted with MS, and doing a whole genome-wide scan, at least 233 gene variants have been associated now with MS. And many of these are important factors that control the activity of immune cell subsets. So many of these gene variants are important for the survival or for the activation of T and B cells, or for their differentiation or maturation into pro-inflammatory types. So that's why, MS not only has this genetic basis, but also the immunological basis as well, because many of the genes encode for elements of the immune system. Next slide, please. Like I said, genes cannot account for all cases of MS, so environmental factors are obviously therefore implicated. And also there are some other data sets that would ascribe a role for environmental factors. So for instance, there's this very curious migration studies data set in which when one locates before age 15 from one area of the world to another, one acquires the risk of MS in that new environment. And in this regard, there are areas with high incidence and prevalence of MS. And I think my next slide shows that, Nitu. So this slide shows worldwide prevalence of MS, and you can see that North America, for instance, and Europe are areas that have very high prevalence of MS. And as you move towards the equator, those are areas with lower prevalence of MS. In the equator, the genetic population is different from that of Northern Europeans or North Americans. So genetics, again, play a role. But in the equator as well, there is a lot of ultraviolet B exposure. Ultraviolet B is what generates vitamin D in us. And that has led to the thinking that vitamin D is an important protective factor against the development of MS. And it turns out that there's a lot of literature showing that vitamin D is an immune modulator. When immune cells are getting activated, the presence of vitamin D can reduce their propensity to be activated. And also other data sets, such as in populations that are properly vitamin D supplemented, there tends to be lower prevalence of MS amongst that population. So again, there's geographical distribution, could be attributed to the genetics, but could also be attributed to the exposure that is in different areas of the world, where in perhaps temperate countries, we might be exposed to certain factors such as viruses that tend to kick our immune system off in an inappropriate manner. But perhaps in the equator, we are exposed to things that protect us, such as ultraviolet B radiation that generates vitamin D in us. Next slide, please. So therefore, the modifiers of MS risk I've summarized in this cartoon, beginning at the top, environmental risk factors, can't really prove causation, but Epstein-Barr virus seems to be an important contributor. Certain pollutants, there's experimental literature showing that certain pollutants can cause immune cells to mature along pro-inflammatory routes. Smoking increases the risk as well as the progression of MS. I don't really know the reasons for that, but in the smoking literature, smokers tend to have immune cells that are more migratory. And so there is a hypothesis that smoking increases risk because immune cells are now more able to cross the blood-brain barrier. And then, of course, we have the genetics, where many of the genes encode elements of the immune system that allows, we think, immune cells to thrive, particularly along a pro-inflammatory route. One may be exposed to these undesirable factors and still be okay if one is properly protected, such as the adequate supplementation of vitamin D. So there are protective environmental factors as well. And then there's also the role of the gut microbiome that we are beginning to get more knowledge of, seems to be able to affect MS development. There is an animal model of MS called experimental autoimmune encephalomyelitis, or EAE, where if mice are kept in a clean facility where they have no gut microbiome, it is not possible to induce EAE in them. Whereas if one now colonizes the gut of those mice with microbiome, and now one can begin to get those mice susceptible to developing EAE. And also from the twin studies in which one monozygotic twin pair has MS and the other does not, there have been studies done in which feces from the affected twin, when used to colonize mice that are kept in a clean facility where those mice previously did not have gut microbiome. So when the feces from the MS twin is used to colonize the gut microbiome, those mice now can succumb to EAE, whereas the feces from the twin that does not have MS is used, there is less propensity of development of MS. So there's a role of the gut microbiome. For me, at the end of the day, MS is a condition of bad luck because you need all of these factors to come together, leading then to the excessive activation of immune cell types, particularly T and B cells along a pro-inflammatory route, where these then enter into the CNS, where microglia are also activated to then produce the pathology of MS. Next slide, please. So medications that I won't be covering today, because MS is an immune-mediated disorder, the therapies that we have to modulate the MS disease cause affect the immune system. And these are referred to as immune modulators or disease-modifying therapies, and they affect various aspects of the immune system, such as deleting T and B cells, preventing T and B cells from being activated, or preventing immune cells from crossing the blood-brain barrier, etc. Next slide, please. These disease-modifying therapies we know are useful. The first one, an interferon beta, was introduced in 1993. So since then, we have a lot of information on the usefulness of these disease-modifying therapies. And we know that for individuals with the relapsing remitting form of MS, where there are relapses followed by periods of remission, and then a next relapse, there are fewer relapses. When relapses occur, lower severity. And when these medications are stopped, MS attacks often recur. So indeed, for an individual diagnosed with MS that goes on these medications, and more often than not, many are diagnosed somewhere between 20 to 30 years of age, although we do have individuals that manifest with MS much later on or much younger. So when one is diagnosed in the 20s with MS and goes on these disease-modifying therapies, they are usually given for life. These days, there are some studies on individuals that have now reached the 50s and 60s that have not had attacks for many, many years. And the question there is whether or not they can be taken off disease-modifying therapies. So there have been studies there, and the data would indicate that some of these patients will have attacks or do have attacks after they stop the medication when they have been on these medications for a very long time. Next slide, please. So here are the clinical forms of MS that I should have introduced you to earlier on. The relapsing remitting form of a relapse, and then a remission, and then with each relapse, the underlying level of disability goes up a little bit. For many of these with relapsing remitting MS, eventually they succumb to a steady progression where the relapses are not that obvious anymore, but where the progression of disability becomes somewhat steady, and that's secondary progressive MS. In about 10% of individuals, the condition begins with disability right from onset that progresses steadily, without much manifestation of an attack in between, and that's referred to as primary progressive MS. The first demyelinating event, be it caught by MRI or by a clinical relapse, is referred to as clinically isolated syndrome. One needs 2 events to become definite MS, and so this first demyelinating event is referred to as clinically isolated syndrome. These days, there's also what is referred to as radiologically isolated syndrome, RIS. So this is an individual, for instance, with migraine that needs an MRI, or suspected brain tumor that needs an MRI, or for whatever other reason needs an MRI, and a spot is discovered. Not quite suggestive of an MS-like lesion yet, but those are referred to as radiologically isolated syndrome. And a number of these will go on to clinically isolated syndrome, where the lesion now or manifestation clinically is more typical of an MS-like attack. And then when they acquire a next lesion, that becomes relapsing remitting MS. So even at radiologically isolated syndrome, in those that with subsequent follow-up become MS, those individuals already have evidence of brain injury. The brain is already undergoing some degree of atrophy, even at radiologically isolated syndrome, telling us that MS starts very, very early on. When we begin to treat MS, a lot of the injury has already occurred, and we are now actually fighting a downhill battle. One challenge is for us to diagnose MS much, much earlier on, and hopefully to intervene from much earlier on. Next slide, please. So another phenomenon then that I want to bring out today is that with the injury, there's also repair, particularly in the repair of myelin. And here I show you in panel A, axons that are coming out at you and the myelin wrapping around. In panel B, demyelination has occurred. And with remyelination, the myelin is put back on. Myelin doesn't tend to be as thick as that in the normal situation, but from a number of studies, we know that this is functional. Next slide, please. So the central nervous system is capable of repair, particularly in the form of remyelination, which is good. And here is a rendition then of, in a normal neuron with its axon that is myelinated. You see 3 myelin segments and a node of Ranvier. In B, immune cells are producing the demyelination. And usually following the loss of a myelin segment, the ion channels that previously were focused at the node of Ranvier now spread out across that demyelinated segment to attempt to still conduct impulse along that axon. And then you have in panel D, remyelination, where the myelin segment tends to be thinner and also shorter as well. This is actually somewhat useful for us histologically because we can then tell a remyelinated from a myelin that has been spared in the first place. So for us, interested in promoting remyelination, the short, thin myelin is a useful indicator for us. From electrophysiological as well as functional studies, we know that these short, thin myelin segments are functional. The example of cats in which the optic nerve is demyelinated, where they lose the ability to see. With remyelination, these cats gain the ability to see. Next slide, please. So remyelination is functional. And here I want to just mention that because the myelin is thinner, now you have less myelin to absorb luxofast blue. If you were doing a histological stain, that area is now less blue. This is referred to as a shadow plaque, which is also a useful indicator for experimentalists who are trying to look at remyelination. Next slide, please. And interestingly enough, in an individual with MS, these episodes or areas of demyelination can be quite close to an area of remyelination. So we see the luxofast blue LFB stain, an area that should be blue white matter area, where you see areas that are totally white, that's demyelinated. We know that that's active demyelination because we do a CD68 stain for macrophages and microglia that are needed to clear the degraded myelin. Those white areas are chock full with these phagocytes. You see those shadow plaques that I've indicated with asterisks. Those are remyelination. And we know that they're not partially demyelinated or undergoing partial demyelination because there are no CD68 positive cells there. So this is how the neuropathologist can tell to some extent an area of demyelination and remyelination. Next slide, please. Remyelination is formed by an oligodendrocyte that matures from its precursor cell. And again, this is supposed to be a video to show you one oligodendrocyte forming extensive amounts of myelin. I won't be able to show you this video, but that is the power of restoring one oligodendrocyte, that it has the potential to form multiple segments of myelin. This is also the problem with losing one oligodendrocyte, where you then lose many segments of myelin. Next slide, please. Here are the oligodendrocyte precursor cells that mature to oligodendrocytes. They're needed to kick the process of remyelination off. And this just shows you that in an MS plaque, we can find these oligodendrocyte precursor cells. So the good news, these cells that are needed for repair are found still in MS patients. And that has led to the realization, next slide please, that many individuals can still remyelinate, particularly early on in the disease course, but that with longevity of the disease process, this repair process is somewhat compromised. The importance of remyelination to restore structure, to enable conduction of impulses again along demyelinated axons. But also there's this important concept that the new myelin protects the underlying axons from further damage, because we know that there are exchange of molecules between axons and myelin. And so they are important to sustain the health of each other. Next slide, please. So here I'm just not going to show you in the interest of time now so that I can still take some questions, that while there are medications that are being tested to promote remyelination, that lifestyle can also help with remyelination. And from work in our lab, exercise, and this is work that is done in animals for which we are trying to get human evidence next. In mice that are demyelinated, if they have access to a running wheel, they are able to generate new oligodendrocytes, form new myelin. Exercise is very important for health, but it's also very important for brain and brain repair. There are a number of mechanisms that are ongoing, but one recent work that we are doing with Dr. Dufour's lab here at the University of Calgary is that a very short period of exercise can change over 140 proteins within the spinal cord. Exercise is just amazing. So anyway, I think I'm going to stop here. The next few slides show us the result of exercise promoting remyelination. I'm going to skip all those. The very last slide just summarizes for us our content today, and that is MS is a condition of neuroinflammation. Many pro-inflammatory cell subsets get activated. They cross the blood-brain barrier, in the process destroying the blood-brain barrier. They produce myelin loss, oligodendrocyte loss, axonal neuronal loss. There is astrocyte reactivity that you will hear about. Through it all, at least initially, the capacity to reform myelin. So with that, I apologize again for the audio-visual problems, and I'm going to go into the chat room and address some of the questions. So again, yeah, apologies for this. I hope you now have some appreciation of what MS is. You will have access to these slides. A number of questions from Chabelle, and questions are great. We really want to encourage lots of questions throughout this series. Those of you who have to sign off because of time, please do so. Otherwise, I'm happy to stay longer with all of you. So just looking at Chabelle's first question, how can we know for sure the age of the lesion? Don't we have the confound of having different rates of lesion formation? Well, it's a very, very good question. So the age of the lesion, that particular slide that I showed you had lesions of different degrees of blue, so that those lesions that still have considerable amount of blue with a lot of CD68 positive phagocytes would be a more recent lesion. The lesion also expands, if you like. So if you imagine a blood vessel in the middle, immune cells coming out to destroy myelin in their immediate vicinity. And then as the lesion spreads, the leading edge is going to move out. So increasingly, you will get less and less immune cells in the middle as they spread out. So that can also tell you to some extent the edge of the lesion. Another way in which the age of the lesion can be analyzed is by staining for the type of myelin proteins. There is this, there are a number of different myelin proteins located on different parts of the myelin sheath. You have one that is called MOG that is on the outermost, and then there is MAG that is in the innermost. So if you imagine a phagocyte such as a macrophage or a microglia that is stripping, clearing, if you like, the myelin debris, if that phagocyte still has a lot of MOG, which is on the outermost part, then that's a more recent lesion. Then another lesion in which the phagocyte has MAG, which is normally on the innermost segment of myelin. So if that lesion has phagocytes that has MAG, but not MOG, which is on the outermost, that is a later lesion. So you can use tricks like this to analyze. And then second question from Chabelle, are there candidates for the viruses? Epstein-Barr virus, top notch in that regard. And then going on to the third question, with the necessity of having microbiome to induce EAE, and given the inherent variability between microbiomes of mice between labs, isn't it hard, therefore, to combine the different findings from EAE models? Yeah, this is a very challenging thing. I am not an expert in the microbiome. You will get a lecture on the microbiome from one of our neurologists, Carlos Camara Lemeroy. I'm learning that there are over 1 trillion microbes in our gut microbiome. So how do you know which ones of these are more critical than the other? Certainly there are classes that seem to be more important than other classes, but as far as I can tell, it's very, very complicated. There's also an approach in MS to use probiotics that you can buy from the pharmacy, where probiotics, as you know, are microbiome. There have been a few trials, small trials that have tested some different probiotics that seem to have some degree of benefit. But the challenge there for investigators is to know which type of probiotic to use. And that is another challenge. Those are all good questions. Are there any other questions from others? We want to really encourage you to have, to have questions. I mean, all questions are great questions, and thinking about questions and sharing questions is also a part of the learning process. So, here's a question from Poria. What are some of the key limitations to remyelination? So here I'm assuming that the question is, what are some of the key breaks or impediments or some of the factors that prevent successful remyelination? This is a very good question. You will also get a lecture on remyelination. Some of the limitations include the following. First, the axon has to be healthy. In long-standing lesions, if the axons have been compromised for a very long time, they can lose their responsiveness to the oligodendrocyte process that is coming to try to enwrap that segment of the axon to form myelin. Sometimes they can even be repulsive. So the axon has to be conducive. And then there are also inhibitors that are present in lesions. There is an inhibitor called LINGO-1, LINGO-1. LINGO-1 was discovered by a pharmaceutical company called Biogen that is very big in MS therapeutics to be an inhibitor. So Biogen went on to find monoclonal antibodies that can neutralize LINGO function, to try to neutralize that inhibitor. And that is actually in a phase 2 trial to try to overcome this inhibitor for remyelination in people with MS. In our lab, we describe another class of inhibitors referred to as CSPGs, and we're also trying to develop tools to modulate that inhibitor. So you have axons that need to be healthy, you have inhibitors in the environment that need to be cleared, and then sometimes you don't generate enough oligodendrocyte precursor cells, or you can't get these oligodendrocyte precursor cells to mature into oligodendrocytes. And there's certainly MS cases from autopsy in which there are inadequate oligodendrocyte precursor cell numbers, or there are adequate oligodendrocyte precursor cell numbers, but not adequate numbers of oligodendrocytes, as if their maturation were impeded. So those are also other limitations if you can't get those cells to populate and then to mature into oligodendrocytes. So and then inflammation, continued inflammation is also a problem. In MS autopsy studies, some of the remyelinated plaques are attacked by new bouts of inflammation. So we still have to control inflammation. Thank you, very good. Yes, any other thoughts? If not, we will bring this to a close then. And again, I want to apologize for not having tested this system with Nitu. But I hope that you get a sense of what MS is about. I hope that you have this initial basis now to launch off. I think Nitu is going to make this podcast available soon, and send you the coordinates to tap into this. We are hoping to, that other lectures can also be podcasted as well. So with that, I hope you have a great summer stint, and let's do the best we can. Thank you all, and have a great day.
Thank you.
Take care. Okay, bye now.